Okay, so now we'll get started on the statistics and reproducibility lecture. And so I think this is one of the more important lectures in the class, particularly for understanding kind of what you do once you've done all of this analysis and how you can kind of interpret the results and make some meaningful scientific outputs from it. So we see that we've sort of covered the analysis techniques so far that we're going to cover in this course. And what this will cover is basically doing the statistics and reproducibility analysis on the tools that we've developed up until this point. The next one, we'll look at more dynamic experiments. And so how do you sort of look at videos or time sequences of images and compare them with each other, and what sort of analyses are possible there. And then it will be sort of about scaling up and then specific applications, and then at the end going into sort of more machine learning and deep learning approaches and sort of the connections between a lot of the things we've developed here and those techniques. And so those are in fact very tightly linked together, and a lot of the things that we've developed thus far really map very well to these kind of approaches. So there's quite a few references for this lecture. With all of these slides, they scroll, so most of the things won't fit on one slide, and so it's quite useful to be able to go down and see. The book, again, that we sort of have with this morphometry in R covers a lot of the basic ideas fairly well, and then we have some more specific ones. There's a nice chapter on hypothesis testing, which I think is helpful to read to understand this a little bit better. Google gives a nice presentation on statistics at Stanford that you can watch on YouTube. There's also a good lecture from UC Berkeley on p-value and sort of what that means and how you can interpret it that I think is helpful to look at. Then, of course, correlation and causation and understanding the differences between it, and then some of the visualization things. So, kind of what we've covered previously on this course is sort of these image enhancement, minimizing noise, doing all of these analysis so that we can get ultimately metrics out of the images that we have. And so we can take this measured data from some source and extract metrics that we can start to analyze. And what we cover here is really the link from these metrics to the final outcome or the statistical analysis that we want to perform. And so ultimately you're collecting all of these numbers so that you can say that there's a difference between these two groups, or that this is the effect of something that you're doing to a sample. And the metrics that you have here are just ways of measuring what that effect are, but they don't actually show that that effect exists or give you any confidence that you're seeing a real effect there. So to kind of go back to the course title a little bit, the course title is Quantitative Big Imaging. And what does big mean? And so, of course, there's thousands of different definitions for big, and we have sort of a lecture where we'll cover much more on big data and big data approaches and how they work. But it's not just, or it's not really limited to large amounts of data sets. And so what big really means for this course is that it's sort of ready for big data, or ready for scaling up or doing lots of different analyses. And so it really means that things are done in a disciplined manner, that there's sort of fixed steps. It's easy to regenerate results, and there's no magic. And so anyone, anywhere, can perform the exact same series of steps that you performed and get the same output. And so if you're clicking lots of different things or looking very carefully into images and changing tiny little parameters by a little bit to get the right answer, it's very unlikely that anyone else will be able to get those results. The next part of this is really having everything automated, and so that every step is cleanly linked together. And so you're doing all of your analysis in sort of one tool or framework, and you don't have to start 5 different programs in order to get the result out. That you have sort of one chain that does your whole analysis, and if you change something about your analysis, you can then rerun the whole chain and get the final answer. And it doesn't require that you remember exactly which step you changed and that you have to go back and rerun everything manually. That it's sort of one completely automated approach. And that, of course, means that 100 samples, 1,000 samples, 1,000,000 samples is just as easy as one. And so, of course, it will take longer to run that computation. You'll need more resources, but it doesn't require any more investment on your part. You've basically established the analysis that needs to be run, and you can scale it easily. The last part is really being able to adapt and reuse the analyses that you perform. And so if you have kind of built an analysis that looks at cells, that you can take the same analysis and look at bubbles without completely changing it. That a lot of it is sort of reusable, a lot of it uses the same inputs and outputs, and it's very clear what's going on. So when we look at kind of the objectives that we had originally, so for scientific studies, particularly if you're trying to compare things or analyze a large number of samples, the goal is really to get everything that you have down to a single number. What we've covered before is actually making sure that this number we create represents what we're interested in representing in the sample. And so that's why we have tools like distance map, like distribution, like thickness, like anisotropy, is to make sure that you sort of have some concrete understanding for what you're actually describing in your structures. And today what we cover is how do we make sure this number is meaningful. And so when we've collected this whole bucket of numbers and analyses that we have, how can we compare different samples? How can we show sort of how reliably we can even get this metric? Because when you have a metric like anisotropy that's defined as the longest length over the shortest length, how do you know? That doesn't tell you anything about how accurately you can measure that. And particularly in more complicated analyses, it's much more difficult to actually know what your measurement error is. And so for those of you that come from sort of a more physics background, you know, if you're measuring something with a ruler and you know that your sort of precision there is plus or minus a millimeter, then you can calculate what the propagated error forward would be for calculating anisotropy. That if it's plus or minus a millimeter in sort of the longest length and the shortest length, then you can calculate what that would actually mean for the anisotropy measurement that you have. But if you're looking at something like a 3D image that you've reconstructed from tomographic data, it's very difficult to say what your error in this image is or what your precision is. You can define things like resolution, but getting more detailed than that can be very challenging. And then once you have an image like this and you're trying to measure what would be the error in a thickness map or the mean thickness over your entire image, this becomes almost impossible to measure. So you can't kind of do this approach where you explicitly say these are the steps that you're going through and this is how the error propagates. That you actually kind of not lose the error, but you lose the ability to quantitatively come up with an analytical method for describing what the error is. So basically the kind of things we want to look at is when within a sample or the same type of samples, how consistent are these values that we're taking out? Do they stay the same or are they constantly changing? And then sort of between samples, and how do we see what the differences are between samples and how can we be confident that those differences are actually meaningful? Or what sort of approaches do we have for convincing ourselves and other people that they might be meaningful? And so a lot of the questions that sort of come into this is sort of how do you evaluate your parameter selection? So you have lots of different tools that you can use like filtering, minimum volume cutoffs, neighborhood operations, morphology, these kind of steps. And how do you know which combination of steps works well? How can you evaluate what parameters you used? And we've covered a little bit of that before, but we'll go into more detail with that today. And then how can you kind of ensure that your techniques do what they're supposed to do? Finally, this is a bit tangential to this lecture, but I think it's still quite important to cover, and there's some good reading material on this as well, is about sort of the visualization. And you see that very commonly in science, this gets wildly underplayed, and people simply make the graphs that MATLAB or Python or KNIME or any of these tools spit out, but they haven't really given much thought to how the data should be organized and trying to make a plot that's easy for other people to understand that communicates your results well. So this is sort of the rough outline for what topics we'll go over here. So sort of the motivation, reproducibility, parameterization, this parameter sweeping idea, sensitivity analysis, unit testing, validation, and then visualization. So that's sort of the general flow for all of the things that we'll cover, and they're not entirely linked together, but they cover the same sort of ideas. So if we go back to kind of the original cell image that we had before, that we've shown in sort of these other lectures, where you have this slide with a bunch of cells on it. I think we see it. No, not on this one. That you're able to get rid of the noise by sort of doing and find the cells in lectures 2 through 4. So this was the segmentation, this was the filtering, all of these other approaches. You could analyze the shape using the shape tensor. And so that gave you some sort of description for how this cell looks. You could even kind of separate linked together cells or cells that we're touching using watershed and make even more metrics to characterize the distribution. And so we've come up with a bunch of different numbers that describe the sample fairly well. And we're confident that it's sort of looking at cells and not looking at the background because we've sort of done that check. And now the question is really, how do you do something meaningful with this so that you can say something useful about the samples that you have? So if you look at kind of what are the most common criticisms of scientific work coming from reviewers or sort of external people looking at the work that's been done, it's this confusion between correlation and causation. And so I'm sure most of you have heard of this before, that this isn't a completely new idea for you. And basically a correlation is a statistical relationship. It's very easy to show. That if you have the data, you can show a correlation in 1 or 2 calculations. And causation implies that there's somehow a link between these two things. And so it's very difficult to show, and it's almost impossible to prove. And so most of the time in scientific work, all you can really hope to do is show correlations. And eventually, once you've shown that there's probably not any other way that this could be happening, you can start to imply that there's a causal link between the things that you're showing. But in fact, that step is very, very difficult, and having one correlation is a huge distance away from having this causation understood. There's also kind of the studies that you're doing are very different for showing these different kind of effects. And so correlation, where you're just trying to show that there's this sort of statistical relationship between these two sets of data points, you can do very well in kind of observational studies. And so observational, or sort of this, what you would think looking at a study like this, it tends to look at very large data sets. I mean, so today, most of the observational studies are really kind of in this big data world where you're looking at millions of visitors for a website a day and what buttons they click on. For example, you don't always have a hypothesis at the beginning. So your goal a lot of times is simply to understand what's happening or what people are looking for. And this very rarely leads to causation. And so you don't really have a hypothesis that you're trying to test. You're usually just trying to understand trends and trying to predict sort of the things that are going on. In 100 cake samples, there was a 0.9 correlation between cooking time and bubble size inside of the cake structure itself, but it doesn't show any link. It's just an interesting finding, and this is usually the first step that you sort of establish what these trends are, and then you move on to kind of the more, or the traditional scientific realm. The boundaries between these are getting blurred, but at least traditionally, this was where science happened, where you have sort of a controlled study. And so you have some concrete hypothesis. And so, if you saw this result, for example, the 100 cakes, there's a 0.9 correlation, then your hypothesis would be cooking time affects bubble size. And so then you would move on to the controlled experiment where you then try to cook 100 cakes exactly the same, except the cooking time is different. And so where you really make an effort to control all the other factors that could possibly come in so that you can really isolate one or two variables that might affect the outcome. You sort of have the specifics of the group much more controlled, and this is much more likely to actually lead to causation because, in this group, the 100 cake samples might have had different batter. They might have been made in different countries because, some countries don't have as much heat or use fire, and other countries use gas ovens. And so trying to establish that correlation or that causation would be very difficult because you simply have too many other variables and you don't know what their links might be. When you switch over to this more controlled approach, then you can try to compensate or have studies where those variables don't even come into place. And then you can try to do that analysis to understand really what's the effect of this. So in genetics, or in animal genetics at least, this is very commonly used because you can turn genes on and off. And so you have a wild population, for example, where you simply look at different traits and you try to map out what traits or what genes are responsible or could be correlated to what traits. And that gives you a very good sort of observational understanding of how things might be connected. But then you need to transition to this more controlled approach where you then turn off specific genes in animals and see what effect they have. Of course, with a gene like this, if you have 50 mice with this XYZ gene turned on and 50 with it turned off, and you have an increase in foot size, then you might think that this gene is responsible for foot size. This is a much more controlled study, but it could be that this gene is causing the animals to simply get larger. And so it makes them eat more, and that's why their feet are bigger, because they're actually heavier. And so this still doesn't give you the causation, but it gets you much closer, and it removes a lot of the other factors that you had. So to kind of go to a simple model, I think a lot of these studies can be kind of difficult to follow, and so to sort of make sure you understand it as well as possible and that you're capable of testing it easily yourself, you sort of have this idea of a magic or a weighted coin. And so if you have a coin and you flip it, you would expect sort of 50% of the time it to be heads, 50% of the time to be tails. And so you can there's this magic or biased coin. You can buy it at a shop. And sort of the first question is sort of how many times do you need to flip this coin to prove that it's biased? Any idea?
Probably depends on how sure you want to be that proof.
Right.
I don't know if you could ever prove it. Like
you can never prove it.
Yeah, in your first example, your first slide, it's something you can prove, but you could feel more certain of the situation, depending on numbers.
Right. So one thing is how certain you want to be. What would another factor be? Yeah.
How often you want to try? How much time do you have to try to find out?
Well, assuming you have kind of unlimited time. So the one factor is kind of how certain you want to be. What would the other factor be for assessing how biased, or if a coin was biased or not? Yeah. Yeah. So if it gets heads 50.01% of the time, it's still biased. But in order to be very confident that in fact it's biased, you'll need to do many, many, many more than if it simply is, it's supposed to be heads 100% of the time. Because then if you do 10 flips and it's head every time, the likelihood it would have been that way if it were not biased is pretty low, and so you're kind of done. But if it's 50.01, then you need to do a huge number of flips to see that actually it is biased, but not very much. And so the more biased something is, the easier it is to see there. So kind of the questions that come up with this are sort of if you flip it 10 times and another person flips it 10 times, is it the same thing as 20 flips? If I flip it 10 times, and this might seem ridiculous for you, but there have actually been many scientific groups, professors even, not at ETH, luckily, that have done this. If you flip it 10 times and then multiply the results by 10, is that the same as 100 flips? And does anyone feel comfortable answering why that isn't the case?
Because you are assuming that every 10 flips you are going to have the same results as the 10 flips.
Yeah. So you kind of scale out your results in ways that they can't be scaled out. So to only have one head in 10 flips isn't that uncommon, but if you multiply that by 10, the probability of that event goes way down. And so when you assume each event, you assume that each flip is sort of a random independent event, multiplying your results by 10 completely kills this assumption. And so it's not at all the same as 100 flips, and you can't do it that way. So if you buy 10 coins and want to know which ones are fair, what's kind of the approach that you have to take? And so, has everyone taken a stochastic calculus class before? So half. Okay. I can't. There's no way to cover all the things you would cover in a stochastic calculus course in one slide. But basically, the way you represent this is we have a stochastic variable x, and each flip represents sort of an observation of this variable, and the act of performing the coin flip f is an observation. And so you kind of normally assume in a system like this that you have a fair coin, and that means the expected value of this flip would be 0.5, so it's 50% heads, 50% tails, and that you have an unbiased flipper. And so you have this f, which cannot influence what the outcome of the flip is, or the way that they flip it always will give you the same result, and so it doesn't change it. You also assume that kind of each flip is independent of the others, which means sort of the probability of these two events is simply the product of their probabilities. And you assume that the expected value of the flip is the same as that of the coin. And so that there's no sort of observational bias when you look and see what the outcome is, what you actually write down versus what you see. Boom. And so those kind of are how those assumptions get put down. And of course, if you've taken stochastic calculus, you've covered this in quite a bit of detail, but I think it's important to have sort of a concrete understanding for what this actually means for a specific case, because there's plenty of people who can do this math and solve these problems and calculate what these products are, who don't have an understanding for what actually they're doing. And then when they do a scientific study, they make all kinds of mistakes because they don't realize the link between what they're actually doing and the math that you have here. Boom. And so basically the way you can look at this coin flip is that each flip gives you sort of a small piece of information about the flipper and the coin. And the more flips you do, the more pieces of small information you collect. And so this is kind of the Bayesian approach to these sorts of problems, that this act of flipping and measuring it gives you slightly more information than you had before. So a single flip in itself doesn't tell you anything, and all it does is sort of update what your opinion is about this coin. And so the idea is that sort of the random stochastic variations, if the coin's not perfectly round, if it's not balanced out, or if your hand flipping is a little bit different every time, these tend to cancel out, but systematic variations accumulate. And so that's why sort of this more flips gives you more information. So if it weren't this way, if it weren't sort of this, your variations weren't all random and stochastic, then more flips wouldn't actually give you more information. So you see with cameras, for example, that if you're taking pictures, you can take many pictures and average them together, and you can reduce the noise that you have. And so if you're taking pictures of stars, for example, you can get very low noise images by taking lots of them together and averaging them together. Why might this be a problem?
Stars moving.
Well, the planet's moving. The stars stay pretty much the same spot, right? And so when you're taking a picture from Earth of the stars, Earth is rotating. And so it makes actually really cool pictures because you sort of have star trails and the stars sort of leave little tracks across the sky. But it means that sort of collecting more information and continuing your measurement longer reduces the amount of noise you have from your camera, but it increases sort of these systematic variations, which is the rotation of the Earth that's slowly happening. And so with a biased coin, this systematic variation would then be sort of this uneven weight distribution inside the coin that causes it to be more heads than tails, or whatever the actual outcome might be. And so if we look for what does this actually mean for a real experiment, each measurement tells us something about your sample, instrument, and analysis. And so all of those things are packaged together. You can't really remove a single piece of that information. And more measurements provide more information. And so if we have lots of random stochastic variations, instrument and analysis, that more and more measurements will cause these variations to kind of cancel out or reduce overall. Normally the analysis in this example doesn't have any stochastic variation, but depending on what you do, so in like proton therapy, for example, you usually will do a measurement on a human body, and then you'll simulate what would happen when you shoot protons and X-rays at this body to simulate what happens when you give them radiation therapy. And then, of course, you have sort of these random variations that occur because you're using a Monte Carlo simulation on this data that you've measured. But with all the analysis that we've discussed up until this point, there's really no variation in the analysis itself. So if you do a segmentation with a threshold, if you give it the same input image, you'll get the same output image no matter what. It's only when you change your input that your output changes. But there are some analyses that you can do that are not completely deterministic, where they can change. Given the same input, you can get different outputs. The systematic variations, however, accumulate. And so if you have things in your system, in this, the planet rotating, or your detector getting warmer and warmer so that the noise level is sort of constantly increasing over the time, then these will accumulate and not cancel out. So to kind of move on to the next point, which is really reproducibility, there's two kind of main things that are meant by reproducibility. The first one is sort of the analysis, and the first one's the analysis, and that's the process of going from images to numbers. And so that's probably the easiest thing to make reproducible, because this basically means that your analysis is deterministic, that when someone gives your analysis the same input, they should get the same output. And so from a scientific perspective, what this means is that sort of anyone anywhere could follow the instructions that you give in your paper and get the same result out as you did. This is sort of the idea of having a methods section and appendices in the papers that you publish, is that there's enough information there that someone else is capable of running your entire experiment and getting exactly the same result. For practical purposes, what this means is that you really don't want to have sort of platform dependence. You don't want to have a lot of proprietary or in-house algorithms. So if you read scientific papers, a lot of them will mention this great in-house algorithm they used for getting rid of noise in their images. And that might give them very good results, but it makes the results they obtain completely irreproducible. Because unless you have this algorithm, you can't do the analysis. And so it's always shocking to me that people can keep publishing papers like this, because it's fundamentally completely unscientific, because they're the only ones capable of producing these results. And they don't even explain how they do it. But somehow this is possible. Ideally, if you want people to be able to reproduce your results and you want to sort of bring science forward, then you can't have proprietary in-house things, and you need to share the analyses that you're performing so that other people can use them on their samples and make sure that the results that you get are correct. Finally, kind of this no manual clicking, tweaking, or copying. And so one of the projects that I observed during my PhD was all the results had to be copied and pasted from one tool into Excel. And the person who did this shifted everything by one row. So they pasted it one row lower than it was supposed to be pasted in. And the results of the study were completely wrong. And no one realized this until after the publication had already been submitted. And so all the things that they had there, you know, the animals were in the left column and the bone thickness were in the right column, and they weren't matched up at all. And so every time you have a step like this, of course you could do the same thing in a program, but at least someone could then see. If you have a workflow, someone can see that, okay, there's a mismatch in where you're putting that, and you can change it, and it'll be fixed in the future versions. When you manually copy and paste, no one can actually see you do this step. And so it's very easy, first of all, to make mistakes, or second of all, to have someone change one of the numbers in between so that they get nicer results. And both of those are very unscientific in terms of the final process. And then the last thing is kind of one script or one workflow to go from your image to your result. And so that you don't have all of these different pieces that need to run independently, you just have one flow that does everything. So for the measurements, of course, this is much more difficult. To make sure that this is reproducible, there's a lot of things you don't have control over. You know, you're not building your instrument, you're not making your samples. If some cells are larger than other cells, you can't really control for that variation. And so the only thing you can really do in the measurement sense is to take each measurement that you have multiple times on sort of independent occasions and compare them. And so this is where you need to sort of measure all of your samples, use it at different times when you have lots of variations in the conditions, in the alignment, in the noise, the temperature of your system, and then to kind of show that there's no change in the results or that the things that you're measuring in these samples are consistent. And so you don't want your measurement or the final results from your measurement to be sensitive to how it's mounted or rotated, to how much noise your system produces, or on sort of the exact way you've illuminated your sample in one case. So in terms of reproducible analysis, if you think about sort of one possible way for doing this, that you can kind of have very simple analysis here is a MATLAB script where you have sort of an image file that goes in, a threshold, and then some code that's run on your image. And then if you have some script like this, then you can run that on many different samples, and you can simply change the image name to run it on different approaches. In Java with ImageJ or R, you can do the same sorts of things where you sort of have your libraries and everything in one line so that you can execute one command, and it does your analysis from start to end. So now, what we had here was sort of this idea of measurement, and that you want to make sure that there's not some statistically significant change in the values that you get out when you do this analysis. And so the way we do this is a technique, or one of the ways you can do this is a technique called intra-class correlation coefficient. And it's basically looking at different groups and trying to see how similar these groups are to each other. And so here you have an example with some value that you're measuring, and you have different groups like cats, people, ID, tiger. It's just sort of made up columns here. And this would be an example with sort of low group similarity. So you see the variations here are between 0 and 1, but they're between 0 and 1 for all 4 of the groups. So there's not really a huge amount of differentiation between cats and people in this case. Something with high group similarity would then look more like this. And so this would be, you have these 4 different groups, and you see that the values kind of group or cluster together, and that's something that you then want to show. So what we've come, or what the scientific community has come up with for assessing this is this idea of this intraclass correlation coefficient. And it's this fairly simple formula where you have this SA squared, which is the variance among groups or classes, and this SW squared, which is then the variance within groups or classes. And so it's basically comparing what's the variance sort of between them over the variance inside of them. And so the idea is if you have 1 or 100%, or 1 means 100% of the variation is actually between the groups that you're seeing. And so all of the noise in your system is sort of explained by the differences in your groups. And 0 means that 0% of the variation is between classes. And so for this graph, what would you expect it to be?
Variation is between classes.
Right. So what would you expect the ICC of this data set to be? I mean, you don't have to calculate exactly, but
It'll be low, right?
low. The ICC would be high. Because it's this, yeah. So it's the variation among the groups or classes over the variation among plus the variation between, or inside of a single group. And so if,
So among is from one class to the next.
right, among is out of the whole data set, and this W is within a single, or within each group. And so if SA is really large and SW is really small, then this ICC factor is very high. So it basically shows that all of the variation that you see is between the classes. And so in this system, I think it's 0.9 or something like that. So that means 90% of the variation that you see is actually the difference between cats and people rather than the differences between cats themselves. So it wouldn't be 100%, because if it were 100%, then you wouldn't actually have this spread here. You just have 4 points. If you had 4 points, then 100%, because you don't have any variation between the values inside of your class.
Good question.
Yeah.
Practical question. When you have multiple groups like this, the variation within the class, the SW, is that the average among the different classes, or how do you?
You mean subtract each class?
What's that?
You mean subtract each class,
Oh,
and then you
that's a
get
way
it
of
up.
combining them.
And in most tools, it's simply implemented, so you can run ICC as a function, but if not, you can calculate it yourself fairly straightforward. But the idea is that you get this number that sort of describes how distinct your classes are.
So the SW takes into account that some of the classes might have a large variation and some don't. You've given examples where within the group the variance range is similar. So now that you might have a situation in one group with a very large variation, in one other one has very small
Right.
variation, and this gets taken into account before when you do the SW.
So when you do the SW, of course, if you have a huge group that has a very large variation and a very small group that has a very small variation, it will weight the large group heavier. And so most of the time you're doing it in setups where you're actually, you try to do this on the minimal set of data that you're interested in. So for example, this one, I don't know what ID is, but that would be something you would probably leave out because you don't want to have that as part of the variation, and it's not actually something you're trying to compare. But if you're just trying to compare cats and people, then you can leave out the last 2 columns because it just makes your analysis more confusing and less easy to interpret this ICC factor. And so if you do have large changes in the order of magnitude of sort of these parameters, then you probably have to redefine your groups a little bit so that the ICC makes sense. But yeah, so if we go back to this original sample, this ICC is negative 0.02, and that's just because of the way it's being estimated in the system. You don't really have to worry about how that exactly gets calculated. Of course, the ratio of two variances cannot be negative, but the result it gives out is. And then if you have a really sort of closely grouped system, then you have this 0.97. So not 100%, but 97% of the variance is explained in the differences between groups. So we can do this kind of analysis for coin flipping, where we actually have 100 coin flips, and we want to see if we can tell the difference between a biased coin and a normal coin. And so here we have a coin A and a coin B, and this is a jitter plot. And so rather than when you have lots of points that fall in the same exact position, like in this case, false, true, coin A, coin B, you can't plot them easily by just showing one point. And a jitter plot adds noise to the plot so that you can see kind of how many points fall in this area. You can, of course, also show it by having colors or intensities or densities or other metrics, but the jitter is one of the very easy ways to show this because it gives you an idea without making it too much more complicated to interpret your data. And so here we have a coin A and a coin B, and we have 100 flips. And basically this, what would the ICC factor tell us in this study?
How much difference there is between two points in terms of their variation. So if you're very low value, that means that there's little variation.
So a very low value means very little variation.
It will tell how
Very little
differentiate
difference.
bias is different.
Yeah.
If a high ICC value meaning will mean that there is a high difference between the bias of the
So a high value would mean that these two coins are definitely different, and a low value would mean that they're the same. And so you can see from this assessment, sort of a value of 0.25 is low, but it's not that low. And so if you're trying to compare these two coins, this doesn't really tell you anything, except for that you need to perform more flips in order to be confident or in order to be able to make a statement about what the difference between these two coins is. So that they're so similar, it's hard to tell. If you then increase it, you can then see, for 20,000 flips, that the ICC drops down to 0.22. And visually, we see that there is sort of a difference between these two groups. And so the coin B got less heads than coin A. And so how would you interpret this? Yeah.
Well, what would you think coin B is false is lower than it should be for a non-magically weighted coin?
Right. So that's the first thing that you kind of see from this result. That's what this ICC tells you, because you'd expect this ICC to be 0 after doing 20,000 flips, because you expect all the random variation that you have to kind of canceled out. And so if it doesn't go to 0, it's an indication that either your measurement or your system that you're looking at isn't actually the same. And what would be another observation you would have for this? So is ICC a good metric for showing that your coin is biased?
Which is good for comparing two points. One of the coins pointing to its bracket seems biased, but it's hard to say what the ICC is comparing.
Right.
If they both came up, if they were both biased the same way, then you still might get ICC close to 0.
Right. So the ICC isn't actually the right tool for comparing, for seeing how biased a coin is, or for showing that it's biased. And so that's a different set of tools that we use. but they're similar in approach. I mean, they have kind of the same goal. The ICC comes at it with the approach of, are the measurements you're making consistent? You know, if the ICC is 0, then that means that sort of all of your samples are the same, and it's a good way of showing that all of your samples are the same for this analysis that you're trying to do. And if the ICC is very high, then it indicates that the variation that you see is really between your samples and there's no overlap. But you see with something like a coin that this coin is fairly clearly biased. I mean, we see here that there's many fewer false than there are in true, and this is 20,000, so it's a significant number of coin flips. But this ICC still doesn't go to 100% because there's so much overlap between these two groups that it won't show that. And so it's not the right tool for showing that these are in fact completely different. It's a better tool for showing how well the metric you've chosen, which in this case was counting heads and tails, can assess the difference between these samples or not. And so even if we were to flip this 1,000,000 times, it still wouldn't go down to 0. But by 1,000,000, we would be almost 99.999% confident that these two coins were different. Okay, so I think we can take a break here and then move on to the next section afterwards, unless there's any questions on that up until now. Okay.